Assalamu alaikum, dear students. I hope you all are doing well. So today in this lecture we are going to talk about membrane transport or transport across the cell membrane. We all are aware that we have the outer protective covering of the cell. We call it as cell membrane or plasma membrane, right? And we all know that it is a semi-permeable membrane. We have already talked about the structure of cell membrane. If you have not watched that video yet, please go and watch the structure of cell membrane. Right, so we have the cell membrane. Outside the cell membrane, we have the extracellular fluid, and inside the cell membrane, we have the intracellular fluid. Now, suppose we have to move certain substances across this cell membrane. Either we have to take certain substances from extracellular fluid into the intracellular fluid, or we can say from outside to inside the cell, or we have to move substances from inside to outside the cell. Now, this actually process is called as the membrane transport. It is called as the transport across the cell membrane. Across the cell membrane means either we are taking these substances from outside to inside the cell, or we are taking the substances from inside to outside the cell. Now, there are various mechanisms actually through which we can move these substances across the cell membrane. And today in this lecture, we are particularly talking about the passive transport, the first type of the membrane transport, or first type of the transport across the cell membrane. Now, let us suppose here is the zoomed view of the plasma membrane or the cell membrane. And above this cell membrane is the extracellular fluid or outside the cell, and below is the inside of the cell. Now we have to move substances from outside to inside or from inside to outside. And we will see how actually the passive transport is working. Right. Now passive transport, when we are talking about the passive transport, there is definitely active transport as well. Then there is vesicular transport. We are going to talk about all those mechanisms in the upcoming lectures. This lecture is going to be only on the passive transport. So passive transport, when we talk about the passive transport, it is actually going to be the transport across the cell membrane, and for this type of transport, we don't require any ATP. So the first point we have to remember is we don't require any ATP. So there is no ATP utilized for this type of transport. And then second thing we have to talk about, or we have to remember about the passive transport, is it is going to be the transport from higher concentration to lower concentration. Either it is from outside to inside, or it is from inside to outside, but it is always going to be from higher to lower concentration. We're going to talk about all the mechanisms. Now we are going to transport from higher concentration to lower concentration. Right. Now, this passive transport actually is divided into two types. There are two types of passive transport. The first type of passive transport is going to be simple diffusion. So diffusion, we all know. Diffusion is actually the transport from higher concentration to lower concentration. Now, there are going to be this passive transport, we can say it is actually diffusion, because we are transporting from higher concentration to lower concentration. Now, this diffusion can be of two types. The first type we are going to talk about is simple diffusion. Remember, the simple diffusion is actually very simple. Here is the plasma membrane, and we all know that plasma membrane is actually phospholipid bilayer. Okay, this plasma membrane, when we talk about the composition of plasma membrane, it is actually composed of that phospholipid bilayer, and it has the cholesterol in between, and it has all these different types of proteins we are going to talk about in the facilitated diffusion. So simple diffusion is actually the diffusion across through this phospholipid bilayer. When we do not need any protein for the transport of substances. When we are moving the substances, simply the substance is getting diffused through the phospholipid bilayer. That is called as the simple diffusion. Okay, now simple diffusion, again, we do not require any ATP for this simple diffusion, and it is always going to be from higher concentration to lower concentration. Definitely. Now, what type of substances can be actually moved or transported by means of this simple diffusion? The first thing which should come in our mind is very important thing is actually going to be respiratory gases. And what type of respiratory gases? We all know it is going to be oxygen and carbon dioxide. So the first thing which is actually being transported by means of this simple diffusion, the first thing is actually respiratory gases, and that is going to be oxygen and carbon dioxide. Now suppose all our cells, all the cells of our body, they need oxygen. Okay, they need oxygen and nutrients, and they need oxygen to perform various metabolic processes, various metabolic activities. And when we are inhaling that oxygen goes into our lungs, and that oxygen from lungs that goes into the blood, and through blood it is transported to all the cells of the body. And when it reaches the cell, it is actually going to be in higher concentration outside the cell, and inside the cell it is going to be in lower concentration, because we have already utilized that oxygen inside the cell. So when these cells continuously need this oxygen definitely is in higher concentration outside the cell. So this oxygen is simply going to get diffused through this phospholipid bilayer, and it is going to get inside the cell. And when this oxygen is being utilized inside the cell for different metabolic processes, and during those metabolic processes we are actually forming a byproduct, and that is going to be carbon dioxide. So we are forming a lot of carbon dioxide inside the cell. So definitely carbon dioxide is going to be in higher concentration inside the cell, and it is going to be in lower concentration outside the cell. So what will happen is carbon dioxide will simply get diffused through this phospholipid bilayer from higher concentration to lower concentration. So this is the this is the first thing which is actually going to be transported by means of simple diffusion, and that is going to be respiratory gases, that is going to be oxygen and carbon dioxide. Then the second thing is actually we have different hormones which are called as steroid hormones. The second thing, steroid hormones. And steroid hormones, we all know. For example, we have a hormone in our body produced, that is aldosterone, then there is testosterone, then there is cortisol. We have different these steroid hormones. Steroid hormones, they are actually formed from cholesterol. And these steroid hormones are actually lipid soluble hormones. Definitely steroid hormones, they are going to be lipid soluble hormones. Now these steroid hormones, they do not have any receptor on their target cells, on this membrane of their target cells. So these steroid hormones actually have the receptor inside the cell. So they are not going to bind on any receptor on the cell membrane. They are simply going to get diffused inside the cell. Wherever their target cell is, they are going to get diffused into the cell by means of the simple diffusion. So steroid hormones are actually also going to be transported across the cell membrane by means of this simple diffusion. Then the third thing which is going to be transported by means of this simple diffusion is actually going to be very important. This also, it is going to be lipid soluble drugs. Lipid soluble drugs. For example, we are taking any medication, we are taking any capsule, for example. and we are taking any capsule, and that capsule is, for example, a lipid soluble capsule, or it is a lipid soluble drug, and that lipid soluble drug also is going inside the cell through this phospholipid bilayer by means of this simple diffusion. So what we have discussed up to now, that passive transport is that type of transport where we don't require any ATP for the transport. The second thing is that passive transport is always going to be from higher concentration to lower concentration. And this passive transport is actually divided into two types, mainly in two types. The first type is simple diffusion, where we have the substances being transported across the cell membrane simply by getting diffused through this phospholipid bilayer. Okay, and definitely it is going to be from higher concentration to lower concentration. The first thing which is being transported by means of the simple diffusion is going to be oxygen and carbon dioxide, and it is going to be respiratory gases. Oxygen is being transported from outside to inside, from higher to lower concentration by means of the simple diffusion. And second is carbon dioxide, which is produced inside the cell, and that carbon dioxide is also going to be transported by means of the simple diffusion, again from higher concentration to lower concentration. And then the second thing which is going to be transported is steroid hormones. For example, aldosterone, for example, testosterone, for example, cortisol. And then we have the third thing which is being transported is lipid soluble drugs. So all these substances are actually going to be transported by means of the simple diffusion. Okay, now when we talk about this cell membrane, I've already told you that it is actually this phospholipid bilayer. Okay, now the cell membrane is actually, or we can say the simple diffusion actually is it is affected by some things actually. Now this rate, when we talk about the rate of simple diffusion, the rate at which these substances get diffused through this cell membrane by means of this simple diffusion. So if we write here this rate of simple diffusion or rate of diffusion, it is actually dependent on certain things. The first thing is actually going to be the surface area of the cell membrane. Now, definitely, if we have more surface area of the cell membrane, the rate of diffusion is definitely going to be more, because we have enough area for the substances to get diffused through the cell membrane. So the first thing is actually the surface area of the cell membrane. Right, surface area of the cell membrane is actually, we can say, it is directly proportional to the rate of diffusion. The second thing is actually going to be concentration gradient. Concentration gradient, हमने जो है पहले ही बात करी है concentration gradient, because passive transport में we are transporting the substances from higher concentration to lower concentration, and definitely the more the concentration gradient, more will be the diffusion. That means the more the oxygen is present outside the cell, more oxygen will get diffused into the cell. Definitely, very simple. So it is going to be concentration gradient. So rate of diffusion is equal to surface area into concentration gradient, and it is going to be inversely proportional on certain things actually. And what are those things which are actually inversely proportional to rate of diffusion? The first thing is actually the weight of substance. The weight of substance which we have to diffuse through this cell membrane, the more the weight, less will be the rate of diffusion. Okay, and the second thing is, which is inversely proportional to this rate of diffusion, is actually going to be the thickness of the cell membrane. Right, so we can say that this rate of diffusion is actually equal to, it is equal to the surface area of the plasma membrane, surface area of the cell membrane. More the surface area, more will be the rate of diffusion. Then it is concentration gradient. More the concentration gradient, more will be the rate of diffusion. Then there is weight. more the weight, less will be the rate of diffusion, and more the thickness of the cell membrane, less will be the rate of diffusion. So we have talked up to now about the simple diffusion. Now let us talk about the second type of passive transport, that is going to be facilitated diffusion. So the second type is going to be the facilitated diffusion. So facilitated diffusion, definitely it is the type of passive transport. So again, we will not require any ATP for this type of transport. So definitely we will not require any ATP for this facilitated diffusion as well. The second thing is, no doubt it is facilitated diffusion, but it is still diffusion. So it will again be from higher concentration to lower concentration. So it is going to be from higher concentration to lower concentration. Now, it is not going to be simple diffusion. It is not going to be the substances being diffused through the phospholipid bilayer. So it is going to be actually the diffusion which is facilitated by certain substances. And what are those substances? They are going to be these different types of proteins, which are definitely membrane proteins. So facilitated diffusion is actually the type of passive transport which is where the diffusion is actually facilitated by these membrane proteins. Okay, so it is actually this facilitated diffusion, it is actually mediated by various types of channels or carrier proteins. Right. Okay. Now, before we talk about facilitated diffusion, let us add one more point in the simple diffusion. Now, simple diffusion, actually, what type of things are being transported? Definitely, it is going to be lipid soluble things which are being transported, and the substances which are actually non-polar substances, we are going to be transporting that by means of simple diffusion. Non-polar without any charge. Because we know that we have when we talk about the structure of phospholipid we have the head of the phospholipid, that is going to be the phosphate part, and we have these fatty acids, and this is the tail of the phospholipids. So we have the head, we have the tail, and this head is actually the polar part of the phospholipid. It is going to be charged. So whenever any charged particle comes towards the phospholipid, it is being repelled by this polar head of the phospholipid. So we are not going to be transporting the charged substances by means of this simple diffusion. We are going to be transporting the non-polar substances without charge, non-charged substances through this by means of this simple diffusion. Now, through this we are going to be transporting the water soluble substances, because definitely lipid soluble substances we are transporting by means of simple diffusion. So we are going to be transporting the water soluble substances, we are going to be transporting the charged particles by means of this facilitated diffusion. But remember, it is going to be again from higher concentration to lower concentration. Now let us talk about all these proteins, and we are actually going to talk about the different mechanisms of the facilitated diffusion. Okay, so let us talk about the first one. The first one is actually going to be, the first type of this facilitated diffusion is actually going to be osmosis. Okay, so osmosis is actually the transport of water. Okay, water transport across the cell membrane. And we know that osmosis, when we have, for example, we have more water concentration outside the cell, and we have less water concentration inside the cell. So definitely water will start moving from higher concentration to lower concentration, and water will definitely move through this channel from higher concentration to lower concentration. Right. And one more thing is that we have solutes present inside the cell. We have solutes present inside the cell membrane. We have solutes present outside the cell membrane. And osmosis ke liye there is one more rule actually. First is water will start moving from higher concentration of water towards the lower concentration of water. The second thing is water will also allow to move from low solute concentration to high solute concentration. Okay, it is solute concentration, not the water concentration. Water will always move through these channels from high water concentration to low water concentration, but water will also allow to move from low solute concentration to the high solute concentration. For example, we have the solutes present here and we have the solutes present here which are actually in lower concentration, and we have solutes present here in higher concentration. And what are these solutes? Most importantly, it is going to be the sodium and glucose. So what do I mean by that? If we have, for example, water, we have to move water across the cell membrane, and water will always move from higher concentration of water towards the lower concentration of water, but water will also allow to move from low solute concentration to high solute concentration. What does that mean? If sodium and glucose is more inside the cell as compared to outside the cell, water will start moving from outside to inside. And if, for example, sodium is more outside the cell, glucose is more outside the cell, water will start moving from inside to outside. So this is going to be the first type of facilitated diffusion, and that is going to be osmosis. And what are these channels actually? These channels which are these channels which are actually particularly for this transport of water, these channels are going to be aquaporins. Aquaporins or aquaporin channels. There are different types of aquaporin channels. We are going to talk about these channels in the upcoming videos. Right, so these channels which are particularly for the transport of water, but remember water will move from higher water concentration towards the lower water concentration, and that is going to be the osmosis. Osmosis, the first type of the facilitated diffusion. Then the second thing is actually we have this another type of channel here, and this channel is actually going to be called as the leaky channel. Leaky channel. Now this leaky channel is kind of leaky. It is always going to be allowing that substance to pass through, and that substance, this leaky channel is actually meant for. For example, it is going to be, for example, sodium leaky channel. Okay, it is going to be sodium leaky channel. There is definitely potassium leaky channels as well. So sodium leaky channel, for example, it is sodium leaky channel, and it is going to be leaky for this sodium. So it is always going to be allow sodium to pass through. Now we all know that sodium is in higher concentration outside the cell. So definitely sodium is in higher concentration outside the cell, and sodium is in lower concentration inside the cell. And we have more sodium outside the cell, we have less sodium inside the cell, and there is leaky channel for this sodium. Definitely it is going to be transporting sodium from higher concentration towards the lower concentration. Right? So it is going to be leaky channel. And the best example for this leaky sodium channel is definitely going to be the cardiac cells, the nodal cells in fact. And another example for this leaky sodium channel is going to be the neuron. Right? We have the leaky sodium channels, for example, present in the neuron, and we have leaky potassium channels as well there in the neuron, and those leaky sodium channels, leaky potassium channels are actually maintaining their one thing that is called as resting membrane potential. We are going to talk about this resting membrane potential when we will talk about the neuron. right? So in neuron we have these leaky sodium channels, we have leaky potassium channels, and leaky sodium channels will allow the sodium to come from outside to inside, and leaky potassium channels will allow the potassium to go from inside to outside, because sodium is in more concentration outside the cell and potassium is in more concentration inside the cell. So that is going to be the second type of the facilitated diffusion, and that is going to be this leaky channels. Now the third thing is actually, now third, this channel is going to be the voltage gated channel. Okay, voltage gated channel. Voltage gated channel. Now this voltage gated channel is actually, so this leaky channel is not sensitive to anything, it is always leaky, right? Now this voltage gated channel, as the name indicates, it is going to be sensitive to any particular voltage. For example, we are again taking the example of a neuron. When we are actually generating something in the neuron called as graded potential, and that graded potential is actually when we are reaching that threshold potential, and that threshold potential is actually going to open the particular channels in this part which is called as axon hillock, and these channels here are called as voltage gated sodium channels. It is again sodium channels. So voltage gated. Now that means in this part, when we are generating a voltage that is minus 55 millivolt, that minus 55 millivolt is going to open these channels, and a lot of sodium will come inside, and that will generate that action potential. In the neurons, to generate an action potential, we need to open these sodium channels, and these sodium channels will only open when they will be hit by minus 55 millivolts. Right, so that means that minus 55 millivolt is a voltage which is actually opening these sodium channels. So these sodium channels become the voltage gated sodium channels, which are actually sensitive to that particular voltage, that is minus 55 millivolts. Right, so we are opening these sodium channels, and that is actually needed for the for generating that action potential. So what did I tell you? That these leaky channels in the neuron, leaky, whether it is leaky sodium channel or it is leaky potassium channel, both are very, very important actually for maintaining that resting membrane potential in the neuron. Okay, and then we have the voltage gated channels. These voltage gated channels are, again, the best example for them is the neuron, and this neuron this neuron actually has these voltage gated sodium channels, and when these voltage gated sodium channels are opened by this minus 55 millivolt, these sodium channels will open and it will generate the action potential. So this voltage gated sodium channel, again, the sodium will move from outside to inside. That means definitely it will move from higher concentration to lower concentration. So minus 55 millivolt, for example, we are hitting, and it will actually bring the sodium from outside to inside the cell. Right? Fine. Now we have another type. So up to now we have talked about facilitated diffusion. First type is osmosis. The second type is leaky channel. The third type is voltage gated channel. Now another type is actually going to be the ligand gated channel. Ligand gated channel. Now ligand gated channel, as the name indicates, that it is going to be opened by a particular ligand, and this ligand is going to be any chemical substance, for example a hormone, for example a neurotransmitter. These are the best examples for the ligand. Right, now ligand gated channels, right, this ligand gated channels के लिए the best example is neuromuscular junction. Right, so we have neuromuscular junction is definitely going to be a junction between a neuron and a muscle. It is going to be a neuron and a muscle. And actually when we have to stimulate any particular muscle, that is definitely going to be stimulated by a neuron, and neuron will actually release a neurotransmitter. Right. For example, this is a skeletal muscle. We have to stimulate this, for example, and this muscle actually will be stimulated by a particular that neurotransmitter, and that neurotransmitter in this case is going to be acetylcholine. Right, and that acetylcholine is actually going to bind on these particular channels, and these channels are going to be ligand-gated channels. Ligand in this case is acetylcholine, and that acetylcholine is going to open these channels. Right, now ligand-gated channels here, for example, acetylcholine will come and bind on this little pocket there, and acetylcholine will actually, when it will bind with this binding site there, it is actually going to open this lid. You can see here it is going to open this lid, and it is going to open this channel. And in neuromuscular junction, it is particularly the ligand-gated sodium channel. Right, it is again going to be sodium channel, and sodium from higher concentration will start moving towards the lower concentration. So again, we have moved sodium from higher concentration to lower concentration, but in this case, this sodium has been moved by means of that ligand, and that ligand is opening this particular channel, so we call it as ligand-gated channel. And in this case, it is moving the cations, that is sodium, so it is called as ligand-gated cation channel. Okay, so that is going to be this ligand-gated cation channel. And then the next type of this channel or facilitated diffusion is actually going to be mechanically gated channel. It is going to be mechanically gated channel. Very simple, it is going to be a channel which is actually sensitive to any particular mechanical stimulus. For example, if I am applying a lot of pressure on my hand, a lot of pressure, for example, something very heavy I am putting on my hand, so that means that will start pain in my hand, and that pain is actually going to be because of the activation of the nociceptors in my hand, in my skin. And those nociceptors, we can also call them as pain receptors. So I have pain receptors in my hand. If I'm applying a lot of pressure on my hand, that is definitely going to activate those pain receptors, and those pain receptors will actually generate that action potential, and that will send that signal towards my brain, and then I will start perceiving that pain. So it is actually going to be those pain receptors, actually that pressure is a mechanical stimulus, and that mechanical stimulus is actually going to open certain channels there. We call them as mechanically gated channels. For example, mechanically gated sodium channels. And when I am opening those mechanically gated sodium channels, sodium is going to go inside these receptors, and these receptors are going to generate the action potential. They are going to generate that impulse, and I am perceiving the pain. So it is going to be mechanically gated channel, and mechanically gated channel because of any mechanical stimulus. If I am opening this channel, for example, it is again mechanically gated sodium channel, and this sodium is actually going to get inside, and it is definitely again important in those pain receptors, for example, pain receptors. And these pain receptors, when sodium will go inside, it will generate actually the action potential there. Action potential, and that action potential ultimately is going to be going through the pain pathway. That is actually going to start that pain pathway from the pain receptors, right? So that is I'm very sorry. That is going to be the mechanically gated channel, again it is the transport of sodium from outside to inside, so it is going to be the facilitated diffusion. And now the last thing is actually it is carrier mediated channel. Carrier mediated channel. So carrier mediated channel is actually going to be particularly for, most importantly, for the glucose. Okay, for example, we have a particular channel in different cells of our body. We call that as GLUT4. That is going to be glucose transporter type 4. Right. Now this glucose transporter type 4 is actually going to transport the glucose. For example, we have, when we are eating the food, that means glucose concentration is going to get higher in our body. It is going to increase in our body. So that means outside the cells, glucose level is going to be more, glucose concentration is going to be more. So it has to be utilized by the cells. So that means we have to increase that glucose uptake. Right? That means we have to take this glucose from outside the cell inside the cell. So suppose we have increased glucose concentration here outside the cell, and we definitely have low glucose concentration inside the cell. Now we have to transport this glucose from outside to inside, right? From higher concentration to lower concentration, and this is actually going to be carrier mediated facilitated diffusion. Carrier mediated facilitated diffusion. And we all know कि जब हमारी body में जो है, when there is hyperglycemia, when we have taken the food, for example, we have taken the carbohydrate rich food, or we have taken the food, we have taken the meals, and after that what happens is glucose concentration is going to go increased, or we can say there is going to be hyperglycemia. So what happens is, by that hyperglycemia in our body, there is going to be insulin released by the pancreas, and that insulin is actually going to increase the number of or concentration of these GLUT4 receptors, so that we can take this glucose from outside to inside. And that is why insulin is very important to decrease the glucose level whenever it is high. It is actually going to increase the glucose uptake by the cells, and it is going to increase the glucose uptake by actually stimulating these cells, so that these cells will start making more of these proteins and will start expressing these proteins in the cell membrane, and so that we can take this glucose from outside to inside. We can take this glucose from higher concentration to lower concentration. And this type of transport is actually going to be the carrier mediated facilitated diffusion. So we have talked about the passive transport. Let us quickly revise this. We have talked about we have talked about the passive transport. And passive transport we have seen there are two types of transports. First is going to be the simple diffusion, where we are transporting these substances simply by diffusing through the phospholipid bilayer, and it is going to be the non-polar substances. It is going to be the lipid soluble substances, and definitely it is going to be from higher concentration to lower concentration, and where we do not require any ATP. Right. Now what are the things which can be transported by means of simple diffusion? It is going to be the respiratory gases, it is going to be the steroid hormones, and it is going to be the lipid soluble drugs. Right, and we have also talked about the rate of diffusion is actually directly proportional to the surface area of the cell membrane and the concentration gradient, and it is going to be inversely proportional to the weight of the substance being transported, and it is also going to be inversely proportional to the thickness of the membrane. That means the more the thickness, more the weight, less is going to be the rate of diffusion. And then the second type of passive transport is going to be facilitated diffusion. Facilitated diffusion, again, no ATP is required, and it is going to be from higher concentration to lower concentration, but it is not going to be getting diffused through the phospholipid bilayer. This type of transport is actually facilitated by these different types of proteins. And the first type of facilitated diffusion is osmosis, that is particularly for the transport of water from higher to lower concentration. But remember, this water transport is also actually depending on the number of solutes, and solute concentration, water will move from low solute concentration to high solute concentration, and solutes most importantly are going to be the sodium and glucose. And then we have leaky channels. For example, leaky sodium channels present there in the neuron, which are very important for maintaining the resting membrane potential there. Then we have the voltage-gated channels, which are sensitive to any particular voltage. We can say that they are closed, and whenever they are hit by that particular voltage, they will open and they will start moving the substances. And in this case, it is voltage-gated sodium channel. So definitely sodium will move from outside to inside. And then we have ligand-gated channels. It is also closed, but whenever any ligand comes and binds on that binding site there, that this channel will open and it will also start moving certain substances. And in this case, the example we have given is the ligand-gated sodium channel, which is again present in the neurons in the axon hillock, which will actually help in generating the action potential there. And then we have the mechanically gated channel, which will be opened by a mechanical stimulus. For example, pain receptors. There will be mechanically gated channels, and they will be activated by that mechanical stimulus, and they will generate the action potential. And that action potential, because of that action potential, we will start perceiving the pain. And then we have the last one, that is carrier mediated facilitated diffusion, which is most importantly particularly for the transport of glucose from higher concentration to lower concentration. I hope it is clear. That is all for today. See you in the next class. Allah Hafiz.